Hi everyone, this explainer will examine a computational study by Get, McCreet, and colleagues that models the aging human brain. The research aims to establish a mechanistic link between specific cellular-level alterations and the functional decline we observe at a macroscopic scale.
So, the central research question is this. How do microscopic changes in neurons and synapses translate into the broader cognitive impairments and distinct electroencephalography or EEG signals that characterize brain aging? You see, bridging this micro to macro gap remains a significant challenge in neuroscience.
Here's how we'll walk through the study. First, we'll outline the central problem. Then, we'll describe the computational microcircuit model they used. After that, we'll get into the specifics of the simulated aging mechanisms, present the functional and EEG findings, and finally, we'll look at the model's potential for machine learning-based diagnostics.
Now, we've known for some time that aging involves numerous cellular changes, things like the loss of specific inhibitory neuron populations and alterations to synapses. The problem is, directly observing how these microscopic events cause functional impairments in living humans is, for all practical purposes, impossible. This limitation creates a substantial gap in our ability to diagnose and develop targeted treatments for age-related cognitive decline.
To bridge this mechanistic gap, the researchers turned to a highly detailed computational model. You can think of this model as a digital representation of a human layer 2/3 cortical microcircuit, which is a critical area for cortical processing. Importantly, the model's parameters are constrained by human-derived data on cellular properties, morphology, and synaptic connectivity. And here is a visualization of that model. It represents a baseline microcircuit of a 50-year-old individual. It incorporates the 4 principal neuron types, excitatory pyramidal neurons, which you see labeled as PYR, and 3 inhibitory interneuron populations, somatostatin-expressing, or SST, parvalbumin-expressing, or PV, and vasoactive intestinal polypeptide-expressing, or VIP. The model is built to reproduce realistic neuron morphologies and connection probabilities based on experimental human data.
So with this baseline model established, the study then simulated a 20-year span of aging, taking the circuit from an approximate age of 50 to 70. They did this by integrating 3 principal aging mechanisms into the model, all of which were derived directly from quantitative human experimental data.
This schematic shows the 3 mechanisms they implemented. First, a loss of specific inhibitory interneurons. Second, a loss of N-methyl-D aspartate, or NMDA, receptors on pyramidal neurons. And third, a loss of dendritic spines, which are the postsynaptic sites for most excitatory synapses. Now, let's look at the empirical basis for each of these changes. The first mechanism is inhibitory cell loss. The model incorporated a progressive loss of interneurons based on human transcriptomic data from Chen and colleagues. The most significant impact was on the somatostatin-expressing, or SST cells. These were modeled with a 24.2% reduction over the 20-year period, reflecting their known vulnerability in the aging process. The second mechanism addresses synaptic composition. NMDA receptors are critical for synaptic plasticity. Drawing from data published by Pegasiou and colleagues, the researchers modeled a 50% reduction in NMDA conductance onto pyramidal neurons over the simulated 20 years. And the third mechanism is structural. Based on imaging data from Patanjik and colleagues, the model included a 20% loss of synaptic spines on pyramidal neurons. This change doesn't just reduce synaptic connectivity, it also alters the passive membrane properties of the neurons themselves, which affects how they integrate signals.
Okay, so with these 3 aging mechanisms integrated, simulations were run to compare the middle-aged and older microcircuit models. The objective here was to see what functional consequences would emerge from these specific cellular and synaptic changes without any further tuning of the model parameters.
The simulations revealed a distinct functional deficit. The raster plots, which show individual neuron spikes over time, display visibly reduced activity in the older model. This observation is quantified in the box plots, which confirm that the aged microcircuits exhibited significantly lower spike rates. The baseline firing rate dropped from 0.76 Hz to 0.57 Hz, and recurrent activity after a stimulus was also attenuated.
This reduction in neural activity directly impacted the circuit's ability to process information. The signal detection error, which measures the overlap between baseline noise and the signal response, increased from 14.3% in the middle-age model to 24.9% in the older one. This indicates the aged circuit is less effective at distinguishing a meaningful signal from background neural noise.
A critical validation step for any model is determining if it can reproduce clinically relevant signals. The team generated simulated EEG signals from the microcircuit activity. The resulting power spectral density plots showed clear differences between the two age models. And what's important here is that the simulated EEG from the aged models reproduced key biomarkers seen in human aging studies, even though the model was not explicitly optimized to do so.
When they decomposed the power spectra, the aged model reproduced several robust findings from the human EEG literature. These include a reduction in the aperiodic offset and the aperiodic exponent. It also showed a reduction in the periodic peak center frequency, which corresponds to the slowing of the alpha rhythm that's frequently observed with age.
Now, the utility of this computational approach is that it allows us to dissect the contributions of each mechanism. By simulating each of the three aging mechanisms in isolation, the study could attribute specific EEG changes to specific cellular changes. As this table shows, while all three mechanisms led to a reduced aperiodic offset, only inhibitory cell loss and NMDA loss caused a decrease in the peak alpha frequency. Spine loss on its own did not affect that particular measure.
The finding that each cellular mechanism seems to impart a unique signature on the EEG signal suggests a compelling possibility that one could potentially infer the underlying cellular state of a microcircuit non-invasively, just using its EEG signal. The researchers decided to explore this with a machine learning approach.
So an artificial neural network was trained on the dataset of simulated EEGs. Its inputs were the EEG biomarkers we just discussed, the offset, the exponent, peak frequency, and power. The network's task was to classify which underlying cellular mechanism, or combination of them, was responsible for producing that specific EEG signal.
The network achieved a mean classification accuracy of 84.1% across all conditions. This result demonstrates that it's computationally feasible to identify the underlying cellular changes with a high degree of confidence, based solely on the emergent EEG signal they produce.
To quote the authors, this work provides a direct link between specific cellular and synaptic aging mechanisms and their consequences for both cortical function and clinically relevant brain signals. This represents a significant step toward a more mechanistic way of understanding age-associated cognitive decline.
This research points toward a future where non-invasive recordings like EEG could be used not just to measure brain activity, but to infer specific changes at the microcircuit level. Such a development could pave the way for more precise diagnostics and, potentially, for targeted treatments designed to counteract the specific cellular deficits underlying an individual's cognitive decline.